probably isn't, there's not enough space for you to draw all this out on this little diagram. It might be helpful if you do it in your notes. So before we go through the details of the Krebs cycle, I wanted to point something out about pyruvate oxidation. So this is a diagram showing you lots of things with lots of arrows connecting to the point that it becomes kind of overwhelming and confusing. But I want you to just focus for the moment on sugars going through glycolysis to become pyruvate, going through pyruvate oxidation to become acetyl-CoA, and acetyl-CoA being fed into the Krebs cycle to release carbon dioxide, all right? And they don't have it drawn on here, but also the NADH. So the thing that I want you to notice is that all the other foods you eat fits into the cycles that we've talked about thus far. So for instance, when you eat fats, you actually break off 2 carbon chunks of the fat to digest, to harvest energy, and you break them off as acetyl-CoAs. So these same 2 carbon acetyl-CoAs that are a product of the pyruvate oxidation are also exactly the same molecules as produced when you degrade fats for energy. So everything else you eat plugs into this cycle at different points. Do you want to see acetyl-CoA? Why not? That's not a very strong endorsement. Do you want to see This is coenzyme A. Does it remind you of anything? It's pretty crazy, right? What's this? Adenine, the sugar, but here it's got an extra phosphate, but then it's a couple phosphates, and then this weird long thing attached to it, right? So I think it should remind you of this, right? Coenzyme A, what's this?
ATP.
ATP. So what recurring theme does this illustrate? What's that?
Small changes.
ATP is everywhere, right?
Small changes.
Maybe small changes result in big biological effect. We really could, I agree, sort of stick it into any one of those categories. But the one that I hope you see is that molecules get repurposed for different things, right? You take ATP, use it for energy, use it for information, you add some stuff onto it to make it into an electron carrier. And now, guess what we do? We use it to attach those 2 carbons and make it a carbon carrier. It's basically the same molecule with this addition on it that's used for that purpose. You, under no circumstances, would be required to recognize this molecule as acetyl-CoA. It's just that we use these terms, and you guys have a pretty sophisticated understanding and recognition of structures of chemicals. And so if you look, you now know, like you can see the similarities in the molecules. And that's why I'm showing it to you. You don't have to memorize the structures at all. It's just to sort of complete the idea. So in the Krebs cycle, we input our acetyl-CoAs, we spit off a couple carbons, and we make a whole bunch of electron carriers. For each acetyl-CoA, we make 3 NADHs, and we don't even talk about this one. I don't talk about it because it's just an electron carrier. It's just another variant of an electron carrier. It's like a different brand of battery, if you want to think of it that way. So we only talk about NADH. Now things get weird, all right? As if they weren't weird enough, now it gets really, really bizarre. So we're going to talk about this process called chemiosmosis, and I'm going to walk you through it, and then we're going to go outside and we're going to act it out. It's okay if when I'm talking about it, it doesn't make a whole lot of sense to you. I think when we actually physically act it out and you move the parts, maybe we can even switch roles. I know that sounds scary since you put so much effort into researching your role. I think we'll come to a deep understanding. Okay. So one thing I realized that I neglected to do up here is I was intending to map out where these different things occur on the diagram, and I didn't do that. So your pyruvate oxidation is also cytoplasmic, as far as I recall. The Krebs cycle occurs here in the matrix of the mitochondria. And this chemiosmosis, this weird process, it occurs right on the membrane that separates the matrix from the intermembrane space. How's that? Good? I know there's lots going on up there, but just remember there's 4 steps for us. You've got glycolysis, pyruvate oxidation, the Krebs cycle, and then this process we're going to talk and act out here. So this is what happens next. I'm not going to write it up there. I'm just going to talk about it and we're going to act it out. The electrons that come in the electron carriers, the NADHs, mostly from the Krebs cycle, but also from a few other parts of the process, donate their electrons to this complex, this protein complex of electron carriers that are embedded in that membrane. So remember how proteins can span membranes? And they can have amino acids that are hydrophobic that let them embed into membranes? Well, there's a whole bunch of these proteins that are embedded into the membrane between the 2 chambers of the mitochondria. The electrons get passed from NADH, turning it back into NAD+. Once it's NAD+, it's free to diffuse back over to the Krebs cycle enzymes and be made back into NADH. So just like ATP can cycle back and forth, ADP to ATP, NADH can go to NAD+, and then back to NADH, back to NAD+, over and over and over again. So one molecule can shuttle enzymes countless times. So the electrons, you can kind of think of them like, have you heard of the game Hot Potato, where you have something hot in your hand, you pass it, because if you hold it in your hand too long, it burns you? All right, well, you can kind of imagine that the electron carriers are like you playing that game, and that electron has energy. And so when it's passed to them, they kind of pass it off to the neighbor. But as they do it, some of the energy of the electron is imparted to the protein. And it uses that energy to pump a proton across the membrane. Electron goes to the next neighbor, it pumps a proton across the membrane, goes to the next, pumps a proton across the membrane. And then at the very end of the chain, guess where it goes? The electron. Everybody take a deep breath. Oxygen. All right? So at the end of the electron transport chain, oxygen receives the electrons, and when it receives the electrons, it actually converts it into, along with some protons, into water. So it's here at the end of the electron transport chain where the oxygen really gets consumed in the process. And it has to be there in order to receive the electrons at the end of the chain. No oxygen, no movement of electrons. They have to have somewhere to go. It's kind of like completing a circuit. You have to complete a circuit to get the current to flow. So at this moment in time, if you look back at our equation,